Well, thank you very much for the invite. It's first time in Slovenia. It's a beautiful city, and thrilled to be here. We've heard a lot about Parkinson's disease. I won't go too much into this other than we've mostly focused on the nigrostriatal dopamine system, but we know that the pathology starts systemically, likely in the gut, enteric nervous system, and spreads throughout brain regions. There's been a tremendous amount of focus on how this pathology spreads, and a lot of the focus has been on alpha-synuclein, particularly like prion-like conformations of the protein. But what I want to talk about today is exosomes, the role that exosomes play in the transmission and spread of Parkinson's disease. This is a completely new area for my lab, so everything I'm going to show you today is unpublished. And so this is our approach. We know that exosomes are a way that cells communicate with each other, particularly neurons, and they're formed by multivesicular bodies, and they're around 40 to 60 nanometers, and the release that contains all sorts of things, including miRNA, double-strand DNA, proteins. And so our approach has been to try to model this in culture under a number of conditions and also in animal models. And the way that you can do that is you can expose or modulate one sort of cell culture and take the media out, isolate the exosomes, and expose other cultures to those exosomes to try to model this spread. So we've used a number of types of cells, everything from cell lines to primary neurons, and we typically will isolate, expose, isolate. We can look at the size, the shape, what's in them. And so this is kind of our general approach. We've done a lot of work in multiple cell types, including SY5Ys, SK cells. We've used a variety of different Parkinsonian-related neurotoxins, including 6-hydroxydopamine and rotenone. We know that in Parkinson's disease, pesticides are linked to Parkinson's. These are two of the models that are used. And so I'll just get right into the data. One of the first things we did was look at untreated exosomes and treating them with rotenone and 6-hydroxydopamine, and we can see that we have increased mitochondrial oxidative stress by mitosocs that we can quantify. We can also take the exosomes from these cultures, expose them to other cultures, and we get a whole number of indicators that are pretty typical of Parkinson's disease, which you see. It's not unique necessarily to Parkinson's disease. It's common to a lot of neurodegenerative diseases, but we see increased oxidative stress. Remember, these cells were not exposed to the toxin itself. They were exposed to toxin-treated exosomes. And we see this with the mitochondrial membrane potential is down, mitochondrial oxidative stress is up. We can also look at cell viability. And so the bottom line from this data is that Parkinsonian disease relevant stressed neuronal exosomes modulate mitochondrial functions in recipient cells. You can think of this as what we're trying to model is bystander effect. You have the pathology spreading throughout the brain. And we can look and see what's in the exosomes. We found several specific exosomes, contents, particularly miRNA 30A5P and C5P. And so these pathways, we can do pathway analysis, and some of our top hits were really linked to pathways that are really important in Parkinson's disease, including mitochondrial pathways, inflammation, and cell death. And so we've done this in primary midbrain neurons, we've done it in cortical neurons, and found very similar effects here. And so one of the things that we can do is we can now, to try to prove, well, is this really involved in the pathogenesis? We can look and we can take mimic exosomes, load them with the specific microRNAs that we found to be important, and treat the cells that way. And so we can use overexpression to mitigate a decrease. We can use shRNA to knock it down. And it turns out when we do both of these treatments, we get the toxicity going in different directions, proving that these microRNAs that are regulated in the exosomes that are treated with toxins do seem to be playing a critical role in modulating cell death. And so we moved into rotenone-treated primary neurons. We've moved from cell culture to primary neurons. And the take-home message from this slide is a lot of what we found in the cell lines, which are a little simpler in terms of the neurobiology, we were able to replicate in both the primary cortical neurons and the primary midbrain neurons. And so it was good to replicate that. And this is where we really got interested. We took a rat rotenone model. This is something we've used many years to model Parkinson's disease. We exposed the rat to rotenone, only a single dose. It normally takes weeks to develop the pathology. We wanted to look at very acute effects, what happens right away. So we took the exosomes, and first thing we got was very similar results to what we found in vitro. Many of these specific mRNAs were altered in terms of regulation. They're up, and we found changes in the exosomes in both the CSF and the serum that were somewhat different. It seems the CSF goes up, and then that is finally decreased depending on the time at 8 or 24 hours in the rotenone-treated rats. So a little bit differences between the serum and the CSF. But this is where we really were finding this very interesting. We took the exosomes from the serum of the rotenone-treated rats, and then we exposed these exosomes into cell culture. And it turns out that we got these exosomes were actually toxic compared to control exosomes to cell culture. So now we can take exosomes that were increased and look at the effect, now taking an animal data and pushing it back into cell culture and really trying to translate this back into our findings and explore this further. So I think right now this is really early for us. We're pretty excited about it, trying to model this pathogenic spread, link animal studies. We might even be able to get some human exosomes at some point and take those into cultures. But we've used multiple Parkinson's disease-related toxicants. They alter the distribution and content. And I think this is really mounting data from us and others that show that exosomes likely are important in modulating this bystander effect and pathogenic spread. This whole project started from a visiting scholar from an Indian university that has a relationship with Purdue, where they send PhD students over for a year. And so it's been a really nice collaboration. And with that, I'll conclude.